Biomedical research and drug discovery have long relied on testing with cells on plastic or in animal models. Both often fail to predict what will happen in a patient. Today, 3D cell and organoid systems offer better models of human biology in the lab. Introducing RASTRUM, the revolutionary 3D cell biology platform from Inventia Life Science. With advanced digital bioprinting technology, creating 3D cell models has never been easier. Printing commences once the kit and multi-well plate are inserted. Droplets of cells and matrix components combine to form hydrogels instantly. The hydrogel matrix is carefully engineered with a tissue-matched stiffness and rich biofunctionality to support each cell type, presenting a consistent environment, batch to batch to batch. With exquisite control over 3D structure, the cell models enabled by RASTRUM are limitless, from simple embedded spheroids to complex co-cultures. Whatever the model, RASTRUM delivers a step change in speed and reproducibility. Whether screening drugs or probing fundamental biology, printed cell models are compatible with standard imaging and assay workflows, and cells can be harvested from the matrix for downstream analysis. With an integrated laminar flow system and intuitive workflows, RASTRUM offers a complete benchtop 3D bioprinting solution. Make complex 3D models simple. With RASTRUM, the revolutionary cell biology platform from Inventia Life Science.